Okay, I'll apologize in advance. This week is going to be lecture dense and longer than normal lectures than what you've been experiencing, but this is all kind of necessary information to cover. So now we'll be talking about the absolute refractory period of the action potential. It's a brief 1 millisecond um, period following the initiation of the action potential. And it starts pretty much at the peak of the action potential, and it's due to the inactivation of those voltage gated sodium channels within that millisecond following the initiation of the action potential. So we end up with this absolute refractory period because within that millisecond, the sodium channels inactivate themselves, and we have to wait out the sodium channels inactivation to be for the neuron to be able to fire another action potential on top of that. So the absolute refractory period starts at the peak of the action potential and goes all the way to the repolarization as the membrane returns to rest. So once the membrane returns to rest and it's over, it's during this time, there's just no way for another action potential to be fired. And it's like if there's another stimulus coming in the cell during this absolute refractory period, that during that peak, and the repolarization, then it's impossible for cells to fire another action potential. And so this absolute refractory period is going to keep the action potential from happening in the same place at the same time on the axon. And so neurons are essentially limited to firing at most once per millisecond, so at maximum 1,000 times per second. This image here doesn't completely do it justice. The absolute refractory period doesn't start right here at the initiation of the action potential. It starts more so at the peak of the action potential when the sodium channels are inactivated. So an absolute refractory period makes it so that an action potential moves in a particular direction. Because we have these voltage gated sodium channels along the length of the axon, starting at the hillock, we can open up gates to start that domino effect, and that action potential will just go in that direction. Or a current could be injected down the middle of the axon, right in the middle, in which case you're knocking down dominoes from the middle, and you're going to get the action potential to go in both ways. If we initiate the action potential at the axon hillock, the membrane depolarizes, and its depolarization starts from the hillock and goes all the way to the terminal. And there's no other direction of movement possible on this, because it's going to spread wherever we have those voltage gated channels on either side of it. But those channels are going to inactivate as the membrane depolarizes. So you can initiate an action potential at the axon terminal by depolarizing the membrane at the terminal, in which case it will open up voltage gated sodium channels at the terminal, and it will propagate back to the cell body. So there's that's just another totally valid way that cells do communicate, going from the terminal all the way to the cell body. And this is also how we're going to keep an action potential going in one direction rather than spreading back and forth at random once they've opened up these sodium channels. So we don't have the backward spread. So absolute refractory periods are this brief inactivation of sodium channels. It's like a domino reset that allows for, that like follows after you knock down a bunch of dominoes. If you want to see them fall down again, you got to reset them back up. You got to pick them back up and line them back up. And so the same idea with these sodium channels is you kind of have to wait for them to reset before they can open again and get reinitiated. So for that brief millisecond, as the membrane is returning back to rest during that repolarization, and it's coming back to resting membrane potential, during that repolarization, it's kind of where we're resetting the voltage gated sodium channel so that it's possible to initiate another action potential after that resting membrane potential has been reached. If there's a stimulus that's coming in, so that during that first action potential, it's going to make it so that that first action potential can go in that one direction and not necessarily go back and forth. And so it kind of forces that directionality of the movement. So the absolute refractory period has a couple of features that give axons a little more specificity in how they're communicating. The relative refractory period, more or less, happens during that after hyperpolarization of the membrane. So after the membrane has returned to rest, there's this overshoot where the membrane's more hyperpolarized than it was when it was at rest. And this is called after hyperpolarization, or AHP. And you can see it in that yellow in the graph at the top. And during this time, it's a relative refractory period because the cell can still fire at this time. It's just relatively harder to fire. It can still initiate an action potential at this time. It's just relatively harder than if the cell was back at its normal resting state. When it's hyperpolarized, this relative refractory period, during that relative refractory period, the membrane is further away from the threshold of activation. So the cell requires a more intense input than it would if the cell was just at rest. So to reach that threshold for activation. So let's say, for example, the cell is at rest at -70 millivolts, and it needs about 5 to 10 millivolts of depolarization to hit its threshold for activation when it's at rest. But during this after hyperpolarization state, or during a relative refractory period, it might need closer to 20 millivolts of depolarization to hit its threshold for activation. So it's possible to overcome this relative refractory period by having a very intense input coming into the neuron. So it doesn't matter that we're in a relative refractory period. And this is why firing rate is determined by the stimulation intensity and the duration of the postsynaptic potentials. The longer they're able to generate depolarization in the membrane of the axon hillock, the more likely they are to be able to override that relative refractory period. And so we can't override the absolute refractory period. That's impossible based on the physiology of the voltage-gated channels, but we can override the relative refractory period with a big enough depolarization. So in terms of action potentials, we have the conduction of these action potentials down the axon, and it's an active process and it's a passive process. The active side of it is that it does require sodium and potassium pumps, and the passive process is mediated by those voltage-gated sodium channels and potassium channels, as the passage of ions between them. And the passage of ions between these channels is non-decremental in the sense that voltage-gated sodium channels are studded along the length of the axon, so there's no decay, because as long as these voltage-gated channels exist along the axons, the action potentials can continue to regenerate itself. Sodium rushes in, spreads, and then that spreading sodium coming in is depolarizing the membrane adjacent to it, and now voltage-gated sodium channels next to it on the membrane can open and spread. And this opening and spreading of depolarization down the length of the axon to the terminal, so that depolarization of those voltage-gated channels until they're inactivated. And during this time, only potassium is leaving the cell and no sodium can come in when sodium channels are inactivated. And then after these cells are repolarized, those voltage-gated sodium channels are possible to reopen or reinitiate a new action potential. So those tightly packed sodium channels allow for these waves of depolarization to occur down the axon, and the spreading of an action potential and the direction of that spread depends on where the depolarization was initiated. So if it was initiated in the middle, it will go both ways. If it was initiated at the axon hillock, it's going to go to the synaptic bouton or the terminal, and it's that's referred to as orthodromic conduction. And this is the most common we'll see in this course, coming from the axon hillock to the axon terminal. And that's also referred to as anterograde transmission. And then we can also, so the spreading of that depolarization go in the opposite direction. So going from the axon terminal to the axon hillock, that's called retrograde transmission or antidromic conduction. It's another totally viable way that the nervous system communicates. We'll just see less examples of it in this course. In terms of the velocity of an action potential, a lot of this has to do with the properties of the axon itself. So action potentials travel much more quickly in large diameter axons. So the thicker the axon, the more current it can run through it. Very thick axons include motor neurons and sensory neurons, like our proprioceptive neurons for knowing where our muscles are. And these are some of the fastest axons we have to carry an action potential. They're very, very thick. And so if you think like a fire hose, it can have a ton of water going through it versus a water hose, like a garden hose. And they have different amounts of stiffness, and you'll have less friction in a large diameter axon or a large diameter hose. And so you can flow more current through it more quickly. And so a very, very thick hose is just has less friction, just like a very, very thick axon, and the current can just move faster through it. Also, the myelination of the axon is another way to speed up the action potential. So the larger and more myelinated axons are going to be the fastest at transmitting signals. So you can see here a comparison of a bunch of different afferent nerves. So these are all sensory nerves. The C fiber you can see is very skinny and has no myelin on it. So it's just like that one red string, and it has no yellow myelin on it. And this is a very thin axon, and C fibers are what carry pain information, like throbbing pain. It's kind of carrying your second pain after injury. And this is mediated by C fibers. So you stub your toe, you may first like yelp, and that's like acute pain. And that would be mediated by myelinated fibers that are thicker. And then about 4 or 5 minutes later, you realize like, oh, your foot hurts, and it's kind of throbbing. And that would be the C fibers now carrying this kind of later pain information. It's just a slower signal transmitting because it's actually, even though it's initiated at the same time as the myelinated alpha fibers carrying this information in, it's just a slower signal being transmitted because it's a skinnier axon and not myelinated. So it's not perceived until much later because the axon's slower at transmitting that signal. So in a myelinated axon, what's different is that we have myelin wrapped around the axon, where we would normally have voltage-gated sodium channels. So what we have here instead are kind of a constraint on where voltage-gated sodium channels can be, and they're found on the nodes of Rambier. And so for all of the ion channels and the pumps that would normally be found on an unmyelinated axon, in a myelinated axon, they're all constrained to just those nodes of Rambier, which are those openings in the axon where myelin isn't. So you'll notice there's a node of Rambier, and then right next to it, there's like a kind of an empty space that looks like a hot dog bun. And that hot dog bun would be the myelin. The hot dog would be the axon. Where there's the hot dog bun or the myelin, you're kind of covering up where there could be ion exchange. So the ion exchange really has to occur only at those nodes of Rambier. And so that's where we'd expect the exchange of ions to occur in a myelinated axon at a node of Rambier. So we see at the axon hillock, we have no myelin, and so we can have these this ion exchange occur. Once the axon hillock triggers being open, then we have the ion channels open, and we can have this flood of or influx of sodium through those voltage-gated sodium channels. And in myelinated axon, there's no domino effect happen because we can't just like open the adjacent channel next to it, because there is no adjacent channel next to it if there's a myelinated segment. And so what ends up happening is in that myelinated segment is that the sodium current, which is depicted in red in these images, it comes in really strong at the node of Rambier, and then it kind of passes through the myelinated segment to the next node of Rambier. And it's almost as if the current has somewhat decayed, because you can see the color is a lot less saturated as you move from the node of Rambier through the myelinated segment to the next node. And it's okay because the next node of Rambier has a bunch of voltage-gated sodium channels that are still going to be sensitive to the depolarization that's spreading through that myelinated segment of the axon. And so as long as the myelination is maintained, that depolarization will be enough, the membrane will be enough depolarized to open the voltage-gated channels in the next segment. So an action potential can kind of reinitiate at the next node and the next node. So the ion exchange is only going to happen at the nodes of Rambier. That's what we refer to as saltatory conduction, because the action potential is actually jumping from node to node to node, and saltare just means to jump. And so it's, again, not a spreading action potential in the case of a myelinated axon, because instead it's actually jumping from node to node, or saltatory conduction. And so there is a requirement for energy that is similar in unmyelinated and myelinated axons, because we still need sodium potassium pumps, and We still have sodium and potassium channels. Everything's just kind of sequestered around the roads of non-VA. And so oftentimes you might find a lot of mitochondria clustered around a node of non-VA because they're making a lot of ATP to kind of support local activity of those sodium potassium pumps just at the node. And in unmyelinated axons, the action potential is passive and decremental process because the depolarization, in myelinated axons, the action potential is passive and decremental because the depolarization diminishes itself from the start of the node to the end of the node. So from the start of the myelinated segment to the end of it, you can see that the decay is occurring, or the red comes in at the node very saturated, and then as it moves to the next node through that myelinated segment, it's much less saturated. So we get this kind of decrement in the charge as it moves to the next node, and it's okay because again, it will be enough to open up that depolarization stimulus, but it does technically decay in an unmyelinated, in the myelinated segment, but it is reinitiated at the level of the next node. Not all axons are myelinated. Some axons are traveling very long distances, are in the periphery, like moving from the muscles to the spinal cord, or generally myelinated axons. So although we did just see C fibers that are peripheral nerves, they're not myelinated. There are exceptions, obviously, to the rule, but because there's a necessity for certain fibers to conduct information very quickly, we would expect those fibers to be myelinated. And so we can expect any nerve that's trying to communicate information very quickly or over long distances, then we would probably expect that nerve to be myelinated. And we have both myelinated and unmyelinated cells across both the central and peripheral nervous systems. It's just different types of glial cells that are going to mediate the myelination. In multiple sclerosis, the immune system is attacking specifically the myelin. The immune system seems to recognize the myelin as the protein in the myelin as a foreign invader and starts to attack it. And so it starts to basically degenerate the free myelin, and it causes scarring in the myelin. Multiple sclerosis just means multiple scarrings, and the scarrings are on what should be a myelinated segment of the axon. So the symptom onset normally includes odd sensory and motor impairment. A patient starts to experiencing pain or tingling or transient paralysis in the peripheral limbs, which we know for the most part are going to be innervated by myelinated axons. And so depending on the state of how the immune system is acting on the myelin, multiple sclerosis gets its name from the fact that we're going to get multiple scars found on the axon. And so looking at a normal axon that's myelinated, and then just below it is a myelinated axon that is scarred or has been attacked. And that one section of the axon would otherwise be myelinated, but this part of the axon was never prepared to be out in the real world. And so if there's an action potential spreading down this myelinated axon, it's going to get kind of interrupted in its conduction of its signals when it hits this scar, because there's damage to the myelin. We don't necessarily have ion channels in this space to allow for the exchange of ions. And so instead, we're going to get this kind of passive decay of current out of this section that is scarred. And we won't necessarily get the reinitiation of the action potential at the next node. So it's going to interrupt communication moving between sites, especially for sites that are innervated by myelinated axons. And so it's as if there's like a hole in the myelinated segments here. And so this is part of why multiple sclerosis results in the devastating symptoms that it does. So now we'll be talking about neurotransmission. And here we're looking at the neuromuscular junction. And here the neuromuscular junction is where we have a motor neuron. It's axon terminal, it's terminating on a muscle cell. And so this is where one of the areas where chemical transmission was first studied in the nervous system. And so we use it often as an animal model. The way that chemical transmission works is we finally have this action potential that got triggered. It makes its way down the axon, and then it reaches the axon terminal. And the axon terminals are going to depolarize on the axon terminals. There's another type of voltage-gated channel. These are called voltage-gated calcium channels. And calcium we haven't really talked about up until now, but calcium is in much higher concentrations outside the cell than inside the cell. And calcium builds up, that's on the inside of the cell, generally gets sequestered or stored in the mitochondria or the endoplasmic reticulum and stays trapped unless there's some sort of signal for it's released into the cytosol. But when the axon terminal depolarizes, this allows for voltage-gated calcium channels to open up, and calcium will rush in because it wants in that cell. Even though there's calcium stored inside the cell, there's not any in the cytosol, so calcium wants in and because there's a concentration difference across the membrane of the intracellular and extracellular compartments. So calcium rushes into the cell through these voltage-gated channels. And when calcium is in high concentrations inside the cytosol, it will work to mobilize neurotransmitter vesicles, or the synaptic vesicles that are found in the active zone and the axon terminal, and they're going to mobilize them to meet with the presynaptic membrane. And when they fuse with the presynaptic membrane, the neurotransmitters are going to dump the neurotransmitter vesicles are going to dump their contents into the synaptic space, and then from there, the neurotransmitters are going to bounce around. And on the other side of that synaptic space, there's a very tiny postsynaptic site. In this case, it's a muscle, which is going to act as a postsynaptic site for this motor neuron. And the muscle cell is studded with a bunch of receptors that are going to receive the neurotransmitter to bind to the receptor, open up a channel, and this channel will allow for the influx of sodium ions, lead to the membrane depolarization of that postsynaptic cell in the muscle. And in this case, when that muscle depolarizes, it's going to have contraction. So the neurotransmitters also are going to need to be cleared away from the space, because if the neurotransmitters don't leave, we're going to get this over signaling of the muscle, which will eventually lead to paralysis. So you have to have some way to terminate the signal, or we're going to have enzymes. So we're going to have enzymes also available in the synapse to chew up neurotransmitters or to remove neurotransmitters. And so there's this process to recycle neurotransmitters or remove or degrade neurotransmitters from the synaptic space, so that the signaling couldn't continue in the future. So for synaptic contacts, there's a lot of different types of contacts. The way we talked about them so far in the course is if the axon releases neurotransmitters onto a dendrite, and those would be axodendritic synapses. And this is the most common type of synapse, especially for excitatory input. Axosomatic inputs are when the axon terminal is releasing neurotransmitters onto the soma or the cell body. And so these are much more common for inhibitory inputs. If you recall, these inhibitory inputs are going to have a much more control over the activity of the cell because they're so close to the axon hillock, because they're coming in at the cell body. Dendrodendritic synapses exist where we have two dendrites that are releasing neurotransmitters back and forth on each other. We don't talk about it much in this course, except that these contacts do exist, and this supports some reciprocal communication between those two dendritic sites. You could also have the release of neurotransmitters from one dendrite onto the other, and then. But again, we don't talk so much about this in the class. And as if I remember correctly, so as you continue to study neuroscience, just be aware that there's these other types of communication where it's not just axodendritic synaptic communication. Dendroaxonic synapses are when the dendrite releases something onto the axon. So for example, we'll see with like endocannabinoids, the dendrite releases endocannabinoids onto an axon terminal and controls the release of neurotransmitters from the axon terminal. In axoaxonic synapses is where we have this axon terminal and it's acting as a presynaptic cell, and then an axon terminal is acting as a postsynaptic cell. So with the exoaxonic synapses, oftentimes these are mediating presynaptic inhibition or presynaptic facilitation. Presynaptic inhibition is if the axon that is presynaptic is talking to the other axon, and this communication is going to inhibit the presynaptic cell, or inhibit the postsynaptic cell, or axon terminal. So the presynaptic cell is releasing some neurotransmitter on the postsynaptic cell, which again is just another axon terminal, but it's going to cause that axon terminal to be less depolarized and therefore make less neurotransmitter release and inhibit the amount of neurotransmitter release from that axon terminal. Axon facilitation or axoaxonic facilitation is where that presynaptic axon releases some sort of neurotransmitter onto the postsynaptic axon terminal and causes it to depolarize on its own and facilitate the release of neurotransmitters from the axon terminal. So this is why you can imagine a neuron at rest, but one of its axon terminals is getting axoaxonic input from another cell, and it's causing depolarization at just at one specific terminal. And so we could have release of neurotransmitters at one terminal, but not at the others. The others could remain silent. And so that's just one way we could selectively control for one of the outputs of a neuron. Despite the fact that we know neurons have lots of branching, we can still control just like a single axonal input or output based off of this axoaxonic facilitation or inhibition. When you have presynaptic inhibition, you can see that the postsynaptic cell traces in blue, and you can see a lot less postsynaptic potential if it's activated by this inhibitory input. And you're going to get a lot bigger postsynaptic potential in the facilitation version. So we're looking at directed versus undirected synapses, and the directed synapse is what we're used to when we talk about classical actions of the axon releasing neurotransmitters onto a dendrite. And we have this neurotransmitter release, and it happens in the synaptic space, and the neurotransmitter is close to wherever the postsynaptic cell is. This is the most common that we find in the nervous system. And directed synapses, again, are going to have these connections that are very close together, a very tiny space between where the neurotransmitters are released and where the neurotransmitters are going to be signaling postsynaptically. And in undirected synapse, vesicles are released along the length of the axon rather than just down the axon terminal. And this means that the site of release of these neurotransmitters from the axon might be really far from the site that they're acting. So an undirected synapse, the way we talk about them are oftentimes we just call them varicosities or strings of beads, where the axon appears like a string of beads. And those are the varicosities. There's like bubbles along the axon, and the bubbles are actually what are going to be releasing neurotransmitters along the length of the axon. This is pretty common for monoamine neurotransmitters like dopamine, serotonin, and norepinephrine. They seem to be released along these varicosities rather than from directed synapses. This is also really common in the endocrine system as well. So here we're looking at this cute little cartoon of what a synapse looks like. We have the axon terminal, and looking down the axon, it's full of microtubules. You got vesicles coming transported down the microtubules, make it to the axon terminal, which is full of other organelles that are necessary for energy production in the synapse. And in the axon terminal, the Golgi apparatus is down there. It's going to be important for recycling neurotransmitter vesicles, and the neurotransmitter vesicles confused with the membrane if an action potential reaches the axon terminal, and they've used that membrane, the presynaptic membrane, and those neurotransmitters get dumped into the presynaptic space, and they're going to do some get to some postsynaptic site. So this would be a directed synapse. So that postsynaptic site is pretty much adjacent to where the neurotransmitter is released. Whereas an undirected synapse, the postsynaptic cell might be quite far away. So here we're looking at the actual anatomy of the synapse. So this is a image produced by using something like 60 different methods. When this came out in 2013, we were really excited as a neuroscience community that we actually got to see it was inside the axon terminal for the first time, because these cartoons clearly don't do it justice. And you can see there's lots of different proteins that are necessary for the synapse itself and for mobilizing vesicles and for recycling some of them from mobilizing neurotransmitters. And then again, this is just an axon terminal. And at the very bottom part, you'll notice that tiny little red area at the bottom. That's what we call the active zone, or the place where the neurotransmitters are actually fusing with the membrane. Everywhere else is just a place for preparing things that are going on in that presynaptic space. So in the active zone, is where neurotransmitter release is possible. So you can get the idea. There's a lot more going on in this tiny simplified cartoons kind of suggest. So neurotransmitter molecules are going to get released from the presynaptic cell in response to depolarization of the membrane, and that depolarization of the membrane would allow for calcium to come in. That calcium is going to allow for the fusion of neurotransmitter vesicles with the membrane. Neurotransmitters are going to get dumped into that synaptic space where we're looking at the neuromuscular junction, where neurotransmitters are going. The neurotransmitter at the neuromuscular junction is acetylcholine. It's the only neurotransmitter released by motor neurons. And so that means that the muscle cell that is the postsynaptic cell is going to be full of acetylcholine receptors. And the acetylcholine receptors, you'll notice, are on the motor M plate, or the postsynaptic density of this muscle. And this chemical synapse, there's this tiny space between where there's this release of neurotransmitters, or the acetylcholine, in between the arrival of acetylcholine at the receptor to stimulate the muscles. And so the way that the muscular junction was identified in this process was just because muscles are huge and they're a useful place to look for clues that might be releasing something like acetylcholine that allows muscles to contract. And they basically worked backwards to find that, yes, indeed, something is being released by these motor neurons, and it later was identified to be acetylcholine around 1920. And so now we know these are chemicals that are mediating this transmission, at least in the muscle. So it's also true in the brain. So first, I'm going to just quickly talk about criteria that are used to classify neurotransmitters. So there's more than 100 neurotransmitter chemicals that have been identified. There's lots of different classes of neurotransmitters. They include amino acid, monoamines, acetylcholines, purines. Those are all classical neurotransmitters. And then we have neuropeptides, lipids, and gases. And we'll talk about examples of all of these today. An individual neuron can make from like one to several different types of neurotransmitters, and a lot of that depends on what types of proteins the neuron is going to express. The vesicles are going to carry these neurotransmitters and can hold any combinations of these neurotransmitters, so they can hold more than one type of neurotransmitter within the same vesicle. And so how do we actually qualify a compound as a neurotransmitter to be defined as a neurotransmitter? The presynaptic cell has to contain the chemical plus the mechanism to make it. So it can't just be something that it picks up and spits back out. The cell is going to have to make it itself and then secrete it. There should also be a mechanism for inactivating this chemical. The chemical must be released from an axon terminal where the axon is stimulated. There are receptors for this chemical present on some postsynaptic target cell that when we directly apply this chemical or agonist that mimics this chemical, it should have the same effect on the postsynaptic cells, just stimulating the presynaptic cell. So you should be able to drop the chemical on the postsynaptic cell and get the same effect as you would get from stimulating that presynaptic cell. And if you were to apply something to block that chemical, then you would expect to reverse or inhibit the effect of stimulation. So agonist and antagonist might seem like unfamiliar words. We'll talk more about this as we continue, but just keep these ideas in mind as we move through the criteria that classify some sort of compound as a neurotransmitter, and why certain neurotransmitters are sometimes an exception to the rule. So for neurotransmitter synthesis, we'll be talking about the synthesis of classical small molecule neurotransmitters, and we'll compare that to the synthesis of neuropeptide neurotransmitters and other non-classical neurotransmitter synthesis. So we'll talk about each one on their own. This slide here is looking at the biogenic amines or monoamine neurotransmitters. So these include norepinephrine, dopamine, epinephrine, and serotonin. Serotonin's at the bottom, and then you'll notice dopamine, norepinephrine, and epinephrine are all at the top. It should be noted that epinephrine and adrenaline are synonymous with the terms adrenaline and noradrenaline. Epinephrine and adrenaline are interchangeable terms, and noradrenaline and norepinephrine are interchangeable terms. They're just, we use them interchangeably throughout the course. Throughout neuroscience, they're really just like American versus British terminology. So adrenaline and epinephrine, same thing. Noradrenaline, norepinephrine, same thing. In the central nervous system, we don't really see much adrenaline or epinephrine. It's something that is only released in activation in response to activation from the sympathetic nervous system from the adrenal gland. But you'll notice that the precursors for biogenic amines that make dopamine, norepinephrine, and epinephrine are all the same precursor, which is tyrosine. So tyrosine is an amino acid. We get it from our diet. Tyrosine hydroxylase is the rate-limiting enzyme that's going to convert tyrosine to L-DOPA. And then from L-DOPA, an enzyme called aromatic amino acid decarboxylase is going to convert L-DOPA to dopamine. And you'll notice that dopamine can be further converted to norepinephrine by dopamine beta hydroxylase on the enzyme. An enzyme, and that norepinephrine can act as a precursor to epinephrine. And so an enzyme is needed to convert tyrosine to the precursor L-DOPA. Tyrosine hydroxylase is a pretty useful marker to identify where these neurons are and identifying if a neuron is synthesizing dopamine. If we want to know if the neuron could synthesize dopamine or also making norepinephrine, then we could look for additional enzymes that are for norepinephrine synthesis. So we would look for dopamine beta hydroxylase, which is needed to convert dopamine to norepinephrine. So if I wanted to differentiate a neuron from releasing dopamine versus norepinephrine, I would have to look at which enzymes are expressed. And the dopamine cell is going to have tyrosine hydroxylase, while the norepinephrine cell is going to have both tyrosine hydroxylase and that dopamine beta hydroxylase expressed. That's going to be the best clue to assess if it's releasing norepinephrine instead of dopamine. You'll notice for serotonin that tryptophan is the rate-limiting precursor, and tryptophan hydroxylase is what's going to convert tryptophan into 5-HT or 5-hydroxytryptophan, and this is a serotonin precursor, and finally it gets converted to a second enzymatic conversion to the final product of serotonin. So you'll notice that serotonin is oftentimes shortened to 5-HT, which is just 5-hydroxytryptophan or tryptamine, and you will see it written as 5-HT most of the time, especially in the notes. So the precursor tryptophan, again, comes from the diet. It's an amino acid. It's required to make sufficient amounts of serotonin, and lots of what we eat just naturally has tryptophan in it. So for those of you that are eating high-protein diets, the amino acid intake might be interfering with tryptophan uptake in the brain. And so some people might take a tryptophan supplement to make sure they have enough tryptophan getting transported into the brain. But most people just get enough through their diet, so it's not a complication you have to deal with. Now we're looking at neurotransmitter synthesis for glutamate and GABA. So glutamate and GABA are two different types of neurotransmitters. Glutamate itself is an amino acid that we can get not only from the diet, but also our cells can make it by basically taking the byproducts of the Krebs cycle. So this is a review for Bio 30. The Krebs cycle or the citric acid cycle is something that's going on in the mitochondria to provide substrates for oxidative metabolism. The Krebs cycle substrates come from our diet, and so we see that fats, amino acids, glucose, they all can act as precursors into the Krebs cycle, or their breakdown products can act as precursors into the Krebs cycle. So glucose can be converted to pyruvate through glycolysis. That pyruvate can enter the Krebs cycle as acetyl-CoA, and pushing acetyl-CoA through the cycle will generate electron donors for the electron transport chain. And this allows for the synthesis of ATP in the mitochondria. So in this Krebs cycle, some metabolites can just shoot off and act as precursors to make glutamate. Vice versa, glutamate can act as a precursor to alpha-ketoglutarate to enter into the Krebs cycle. So all cells have mitochondria. That means they're all expressing the enzymes to make glutamate. And so to differentiate a cell that releases glutamate from any other cell, you have to find a different marker than just synthetic enzymes. So we're going to have to talk about how to identify glutamate releasing neurons in a little bit. The other thing to note about glutamate is that once it's in the cell, it can be used as a neurotransmitter or a precursor to alpha-ketoglutarate to enter into the Krebs cycle and provide energy for the cell. But it can also be converted into other byproducts that are also neurotransmitters. So glutamate can get recycled into glutamine, which is another type of amino acid. And the glutamate recycling is commonly seen in astrocytes. Astrocytes remove glutamate that's released by neurons into the synapse, and they additionally are going to convert that glutamate back into glutamine, which is released from the astrocyte and taken back up into the neuron to convert back into glutamate. So astrocytes are saving the day, not only by mopping up glutamate the neurons have dumped into the synapse, but also supporting neuron glutamate synthesis by providing that glutamine precursor. We also see that astrocyte being a superhero as well later. Glutamate also acts as a precursor to GABA. The enzymes for making GABA are only expressed in the neurons that convert glutamate to GABA. So any sort of cell that's going to release GABA will express the enzyme glutamate decarboxylase, or GAD. And GAD enzymatically converts glutamate to GABA. And so all cells that have the ability to make glutamate, that's true. But if the cell additionally has glutamate decarboxylase, then we're going to expect that cell to be releasing GABA because it's going to be converting that glutamate to GABA. And so we can also notice that GABA can be converted a couple other times through enzymatic tweaks, in which case it can re-enter the Krebs cycle and be recycled back into glutamate as well. Acetylcholine is something that's synthesized, again, from precursors available in the diet. Acetyl-CoA is one precursor for acetylcholine. It's at the top of the entry point to the Krebs cycle. Glucose can get converted to pyruvate through glycolysis and then enter pyruvate through the mitochondria, and that will get converted into acetyl-CoA. That acetyl-CoA can go through the Krebs cycle to make substrates for oxidative metabolism, or instead the acetyl-CoA can shoot itself off and be found in the cytoplasm, in which case it can be combined with choline. And choline is something that comes from the diet as well. It's very similar in structure and function to B vitamins. We get it from eggs, meats, nuts. Our liver also makes quite a bit of choline, and choline is used in synthesizing the cell membrane. So acetyl-CoA is pretty abundant in cells. There's transporters in the neuron for it. It's important to have lots of choline around for just normal cell membrane stability. So acetylcholine synthesizing cells has choline and acetyl-CoA. They can be combined, and this is catalyzed by the enzyme choline acetyltransferase, or it's shortened oftentimes to CHAT or CHAT, and to make acetylcholine. And so this is a very simple mechanism. It just requires acetyl-CoA and choline and the enzyme CHAT, and it mashes the two together, and you got acetylcholine. So in terms of the classical neurotransmitter, you'll notice this is, we just need some sort of enzymatic conversion of an amino acid or dietary precursor, and the presence of certain types of enzymes that are going to be expressed are going to dictate what each neuron is going to release. So if a neuron is expressing tyrosine hydroxylase and beta hydroxylase, we can be certain this neuron is releasing norepinephrine and not dopamine. If I know a neuron expresses glutamate decarboxylase, I can be pretty sure it's making GABA. If I know it's expressing CHAT, we know it's making acetylcholine with glutamate. With glutamate, it's something that all cells make because it's an essential amino acid. So for us to actually identify a neuron that releases glutamate, we have to find a different marker in the cell. So to be classified as a neurotransmitter, there needs to be a mechanism for release, which we'll discuss shortly. That release is mediated by the fusion of the synaptic vesicles with the presynaptic membrane. But those vesicles would be releasing empty contents if they're not packaged with neurotransmitters. And it turns out there's transporters on the vesicles that take neurotransmitters and package them into the vesicle so that they can be released by the cell. And so there's a vesicular transporter for glutamate. It's written down here as V-GLUT. And the V-GLUT transporters on vesicles are found on neurotransmitter vesicles that are going to allow for the packaging of glutamate into vesicles. So this is really how we can identify a cell as being a glutamate releasing cell, is if it expresses a V-GLUT. But only some cells are going to release glutamate, and those cells that express V-glutes are going to be the ones that can release glutamate. An acetylcholine releasing neuron won't necessarily express V-glutes, even though it can make lots of glutamate. It's not going to release it unless it were to also express the V-glute transporter. So if an acetylcholine neuron additionally expressed V-glute, then the acetylcholine neuron is going to release vesicles that have both acetylcholine and glutamate packaged into them. So the classical neurotransmitters are in this category because they were discovered first, and they all follow a similar principle. They're all going to produce rather punctate effects, which means they tend to produce responses in the postsynaptic cell that are relatively fast, like EPSPs or IPSPs. And it depends on the type of receptor it acts on, some sort of change in metabolism or gene expression in the cell. These effects are triggered by the release of neurotransmitters acting on a postsynaptic receptor. The receptors are going to dictate the scope of the effects. Classical neurotransmitters are only going to be released with the stimulation of the cell, so with an action potential coming down the axon. Classical neurotransmitters are released when the cell is stimulated, so with the conduction of an action potential and the ultimate depolarization of the axon terminal. This is going to mobilize vesicles to fuse with the membrane so that they can release neurotransmitter contents with every action potential. Classical neurotransmitters that are synthesized by a cell are going to be dumping themselves into that synaptic space. We'll take a look at the cell body of this neuron. There's some expression of genes. Those genes are going to be making enzymes that are required to convert these dietary precursors like tryptophan here into the final neurotransmitter product of serotonin in this case. So that mRNA you're seeing expressed coming from the nucleus is going to be translated into some sort of synthetic enzyme. In this case, it's tryptophan hydroxylase. And it's going to be needed to synthesize the serotonin and all of the other enzymes that are required in the chain to synthesize serotonin would also be expressed by this neuron. Then the neurotransmitters can get synthesized either in the cytoplasm and packaged into vesicles as they're coming from the Golgi apparatus. So in the soma, the vesicles are going to be stuffed with neurotransmitters on their way down the axon, and also at the level of the axon terminal. There's a lot of vesicular recycling, in which case the Golgi apparatus and the axon terminal, which you might notice being down there as well, it's common for neurotransmitters to be repackaged, or classical neurotransmitters to be repackaged in the terminal with the depolarization of neurons. The vesicles fuse the presynaptic membrane, and the neurotransmitters released into the synaptic cleft to act on the receptors, cause a postsynaptic response, and then they can be removed from the synapse and recycled back into a neurotransmitter. So this, or into a neurotransmitter vesicle. So this is true for classical neurotransmitters. So we're going to talk about some less classical neurotransmitter effects. So neuropeptides synthesis, neuro meaning brain, related, and peptide is an amino acid chain or a protein. And so these are proteins used as signaling molecules in the brain. And to synthesize neuropeptides, you need to transcribe genes from the DNA into mRNA. Then we need to translate that mRNA into an amino acid chain. And that is what's going on in that first box at the top. We have transcription of a particular gene. It's going to make this long mRNA chain, and it will be cut a couple times to make this final message so that it can be translated into some functional amino acid chain. That translated amino acid chain that's first made is called a pre-propeptide or a propeptide. And that propeptide is going to get proteolytically processed, which just means that the proteins are going to get cut. So the pre-propeptide or the propeptide chain can be cut by proteolytic enzymes that are found in the cell. These proteolytic enzymes are going to dictate which smaller peptide chains are produced. So we can make a bunch of smaller peptide sequences, and many of the smaller peptide sequences can act as their own independent signaling molecule to be released into the extracellular space and have actions on a postsynaptic cell. So for example, we can see this very long peptide sequence as for POMC or pro-opiomelanocortin. And this can be a very super long peptide sequence, and this would be our propeptide signaling molecule. And it can be released as a propeptide signaling molecule that is just a very long sequence and have actions on its own receptor. But this pre-propeptide can also be cleaved a bunch of times or broken up a bunch of times. And everywhere you see a little color center kind of block on that peptide is a site that can be lysed or cleaved on this peptide chain, in which case we have the options to produce a lot of different peptides for each of these little cleavage points. So a cell is going to make peptides depending on the pre-pro peptide it expresses and the type of proteolytic enzymes it expresses. So depending on what type of enzymes are around to cleave this peptide is going to dictate what types of peptides that the cell releases. So if it's a type of enzyme that would cut the peptide, or won't cut the peptide altogether, or it doesn't express any of these proteolytic enzymes, it's just going to release that full POMC peptide. If it's a peptide that is expressed at the same time as proteolytic enzymes that can cut it, then we're going to get the release of a lot of other smaller neuropeptide molecules that would have their own signaling effects. And so we could have these like individual little peptides that are made from the cleavage of this longer propeptide. But there's, again, going to be specificity of what receptors these different peptide molecules bind to. So POMC is going to have its own receptor, and some of the other tiny molecules that it could be, like AVP, for example, could be, or sorry, let's see, what's part of pro-opiomelanocortin here. So if we broke it up into, like, if we had an ACTH here, that is going to act on its own receptor, or we could release the entire full length peptide of POMC and it can have its own receptor that it acts on. So again, what the neuron is going to release really just depends on what types of enzymes it expresses that could potentially cleave these different portions of the peptide. And if it doesn't express these cleavage enzymes, then we're going to release the full length peptide. So it really just depends on what the cell's expressing in terms of the peptide itself, and then in terms of those proteolytic enzymes, should this be cut further or not? And if it's cut further, we're going to have a different effect than if it was released as a full length. So we can kind of look at now this half of the neuron over here, where we have now peptide neurotransmitters. And we can see this comparison to the classical neurotransmitter synthesis and packaging and release. So with peptide neurotransmitters, they have to be synthesized in the cell body because that's where the nucleus is housed. So we first have to express the gene, the mRNA, transcribe, and then translate it into a long pre-pro peptide. Then we have that long peptide. We see it enters into the Golgi apparatus, and there it can have some level of proteolytic processing that happens to produce those little cleavage products. And then we can see the endoplasmic reticulum is right next to the nucleus. And so it has all those ribosomes there. So it can translate the mRNA of that peptide right there and can leave through the endoplasmic reticulum and find its way to the Golgi apparatus for cleavage of the amino acid chain. And those peptide products can bubble off of the Golgi apparatus and into a little neurotransmitter vesicles. And these vesicles are going to get packaged with neuropeptides, and then also additional enzymes, additional proteolytic enzymes that can continue to cleave peptides to even smaller independent products on their way down the axon. And so this proteolytic processing of a peptide can happen in the cytosol and the cell body, but a lot of it is happening in the Golgi apparatus and the vesicle on its way down to the axon. The neuropeptide vesicles are going to get transported using fast axonal transport down the axon tail to the axon terminal. They'll be released with increasing amounts of depolarization. So with increasing activity of the cell, we're going to see more and more neuropeptide release. So it's a gradual release in response to the cell's overall activity. The release of neuropeptides tends to result in neuromodulatory effects, mostly because the types of receptors they act on are slower to produce cellular effects, and also because neuropeptides have slower degradation and removal that tend to depend on the presence of proteolytic enzymes in the extracellular space. And so they tend to stick around in the extracellular space and diffuse to extrasynaptic areas. And this allows them to have kind of actions that are distal from the site of release. And there's also very little evidence to suggest that neuropeptides recycle themselves back up into cells, since it's been shown that certain peptides can, but it's not necessarily true for most of them. What we do know is that Once they get released in the cell into the extracellular space, most of it bounces around until it finds its receptor. It might run into an enzyme that degrades it in the extracellular space. And it's they aren't necessarily targeting at directed synapses to the same extent that we see with classical neurotransmitters. So we could have like accumulation of neuropeptides outside of the cell, as long as the cell is very active. And so if the cell is very active, we're going to expect it to continue to release neuropeptides. But because the cell isn't able to like regenerate neuropeptides as quickly as it is to recycle and regenerate classical neurotransmitters, so it means for us to get more neuropeptides available for the release, the cell has to continually synthesize these neuropeptides at the level of the cell body. And we have to wait for them to make their way down to the axon terminal, whereas classical neurotransmitters can like have this recycling of their vesicles and repackaging of their vesicles down at locally at the axon terminal as well. So we can really easily increase the readily releasable pool of classical neurotransmitter vesicles compared to what we can do with peptide neurotransmitters, just because we can't easily recycle the peptide neurotransmitters at the level of the terminal. Okay, so we're going to talk about how vesicles are packaged and transported down the axon. So neurotransmitters are going to be packaged into secretory vesicles or synaptic vesicles that are going to fuse with the presynaptic membrane with the depolarization of the axon terminal that comes off with an action potential. And so vesicles and cells are going to originate or bud off of the Golgi apparatus in the cell body. And the vesicular transporters are proteins embedded in the membrane of the vesicle that are required for the packaging of neurotransmitters into their vesicles. So on these transporter proteins are a bunch of, to get these transporter proteins, we need gene expression and transcription and translation on the rough endoplasmic reticulum. They find their way to the Golgi. The Golgi packages them into these little synaptic vesicles, which are little lipid bilayer bubbles that are going to have these transporters built right into them or embedded in the membrane of the vesicle. So you can maybe see it better here. And so any free neurotransmitters that's been synthesized in the cytosol can just find its way in now and directly into the vesicle if there's a transporter for that neurotransmitter embedded on the vesicle. And that cell is indeed synthesizing that neurotransmitter, then we can get that neurotransmitter into the vesicle. And then if there's depolarization at the axon terminal and the vesicles are ready there, then we can have release. So neurotransmitter packaging can happen at the level of the soma, but it can also happen all the way down, all the way on the way down the axon to the axon terminal. And repackaging at the level of the axon terminal can happen for classical neurotransmitters. In terms of the neuropeptides, they're synthesized in the rough endoplasmic reticulum. And they're transported to the Golgi apparatus where they butt off the apparatus synaptic vesicles, and they're already packaged into the synaptic vesicle along with the enzymes that are going to be needed for further proteolytic processing of the peptides. So vesicles, after butting off the Golgi of the soma, will just kind of zoom down using fast axonal transport and get to the terminal to be released with increasing amounts of depolarization that come down the axon. So these are slower to replenish those particular vesicles for neuropeptides because we have to wait for the synthesis of more peptide at the level of the cell body. So it takes time, and we have to consider how far you got to move down the axon. Again, the transporter proteins are really actively pumping the transmitters into their vesicles. And that's what we mean by active, is it actually does require ATP. It's one of the things the brain's really burning energy up on, is packaging each individual neurotransmitter into a vesicle, which is a wild amount of energy if you really do the math for every neurotransmitter that's been packaged. We can see that there's an appropriate transporter for each type of neurotransmitter. V-glutes are for glutamate. V-mats are for monoamines. There's one for acetylcholine. And so we can, again, see if a cell is releasing a particular type of amino or particular type of neurotransmitter by just looking at the type of vesicular transporters that expresses, because that gives us a clue as to what types of neurotransmitters it's packaging into vesicles. In terms of the types of vesicles that our neurotransmitters are packaged with, we have small clear core vesicles that are going to contain the classical neurotransmitters like acetylcholine, glutamate, GABA. So here at the top is a pretty good image of them. It's taken using electron microscopy. So in yellow is the dendrite, orange is the axon terminal, and that's where you're going to expect to see these little synaptic vesicles in the axon. And those small vesicles, like those little bright blue looking balls in the axon terminal, are the vesicles. And then the larger blue bubbles, they're organelles, the Golgi mitochondria. Then there's large dense core vesicles. You might notice more so down at the bottom. Those are those large black looking balls. And the large dense core vesicles are going to contain monoamine neurotransmitters as well as neuropeptides, and they're of a larger diameter. Oftentimes you may not be able to find these large dense core vesicles in certain axon terminals. It may be just because they're being replenished as the cells released a bunch of neuropeptide. So another thing to note in the cartoon and at the bottom of that image here is you'll notice that there's coexistence where there's both large dense core vesicles in addition to small clear core vesicles, all in the same axon terminal. So this is not just something strange about the cartoon. This is a real thing. And this is just called coexistence, where a cell can release multiple types of neurotransmitters. And so we can have multiple types of neurotransmitters either packaged within the same vesicles, or we might have different types of vesicles that are released by the same cell. And so we could have, acetylcholine and glutamate in the same vesicle, as long as we have vesicles transporters on those neurotransmitters present in the neuron itself. If a neuron has this coexistence, it's going to release those neurotransmitters all at once with every single depolarization. So the neuron can't necessarily dictate that it's going to release acetylcholine but not glutamate. If it has the coexistence of both glutamate and acetylcholine, it's going to release both types of neurotransmitters simultaneously with an action potential. And so if that's what the cell expresses and synthesizes, that is what the cell is going to be releasing with every action potential, this mixture. And so whatever a cell releases is a product of what the cell is expressing. So you can picture there's lots of options of what a cell is actually releasing. And if you want to know what a cell is releasing, then you have to know what types of proteins it's expressing. So you'll notice on the slides that there's, or your Google slides, it won't be active on this video, but on your Google slides, this particular image is a GIF. And it's a little protein on the bottom walking along the axon, and it's actually walking along a microtubule inside an axon. This cute little protein is walking a vesicle down the axon terminal using the microtubules, which are laid along the length of the axon. And so the microtubules act like a highway that connect the cell body to the axon terminal. And there's two ways we can transport from the cell body to the axon terminal, or vice versa. If we're trying to move vesicles down to the axon terminal from the cell body, we have to rely on the use of these motor proteins that are going to hang onto the vesicle and like walk it down the highway. So you can look at the GIF to see it strut. And this video also down here, you can link to it in your Google slides as well. It's really just a really cool visual to show how it walks. It has hinges and it moves by literally like walking down the microtubule. It's one of my favorite proteins. So this little motor protein kinesin has two little legs that kind of slam down quick and pick itself up and walk down the microtubule highways. And this movement is going to require energy. It's slow. It's moving about 1.5 centimeters an hour. So you can imagine if we're trying to replenish a pool of neuropeptides in an axon terminal, how long it actually might take, and hence why the cell might just consistently make and express some of these proteins to keep up with demand. So now we're going to talk about how these neurotransmitters are actually released. So the steps in neurotransmission, here's a reminder of what an axon terminal might look like compared to the cute little cartoons. We get a lot of different proteins that are available in the axon terminal, and you'll notice that there's lots of vesicles. One thing I do want to bring to your attention is the red bar between numbers 2 and 3 in that image at the top here. So this red bar right under my hand. And this is supposed to symbolize the active zone of the axon terminal. And the active zone is a site where all the machinery or all of the protein machinery for vesicles are going to bind and fuse to the membrane. And so we're going to expect to find all of the machinery for vesicle fusion in the axon or in the active zone, not in the entire axon terminal. So it looks like there's vesicles everywhere in the axon terminal, but only those vesicles that are actually in the active zone. So those are actually going to be ready to be released. They are in the readily releasable pool. And so vesicles that aren't ready to be released, you can see that they're just kind of further away from the active zone, and they're just either waiting to be mobilized, or they might even be in the process of being actively recycled, but they're certainly not going to be released with the next action potential. So only the vesicles down in that red active zone are going to have the ability to fuse with the membrane with the next action potential that would come. The active zone is kind of blown up down here in this little cartoon. So this is like what you might expect the active zone to have going on in it. And so this is where we're going to expect vesicles to actually release their contents when we have depolarization of the membrane. So the axon terminal will experience this action potential arriving, and the depolarization of that axon terminal will open up those voltage gated calcium channels, which are otherwise closed, and they're gated by the voltage of the membrane. So the membrane voltage depolarizes during an action potential, opens calcium gates, and now calcium can rush into that presynaptic membrane. Increasing calcium is going to get this activation of synaptic proteins that are going to mobilize these vesicles that otherwise would be on like maybe microtubule networks, or get them closer to this readily releasable pool here in the active zone. And once in the active zone, calcium is going to activate additional synaptic proteins, for example, SNAP25 and synaptotagmin and syntaxin, and they're all going to be necessary for fusing the vesicles with the synaptic membrane. And so these proteins are grabbing vesicles that are in this readily releasable pool, bringing them to the dock, fusing them on the presynaptic membrane, and allowing them to dump their neurotransmitter contents into the synaptic cleft. And then once we have exocytosis of the neurotransmitters, you can see that the vesicle now sits empty because the vesicle is empty, its contents out into the synaptic space, or exocytose the neurotransmitters out into the synaptic space. And that empty vesicle is going to get recycled by bubbling itself back off the membrane, either with the help of additional proteins or lipid interactions within organelles, so that it can either be filled back up with more neurotransmitters, or it can be degraded if it's no longer any good. So here's another image of what you can see here with the electron microscope. We see the active zone. The electron micrograph and this cartoon, we can kind of see kind of what they're comparing to in terms of there's an axon, there's a vesicle here bubbling off. And so that would be kind of expected to be what's going on here, where we have these little like blibs or like little circles in the membrane. We can see this exocytosis happening over here, these two vesicles that are like fused with the membrane. And so that's kind of what we'd expect to see with this exocytosis. And from there, you can see that there's this active zone where the vesicles are fusing with the membrane, emptying out their contents. You can see the vesicles that are also left over are bubbling off. And the ones that are still bubbling off or left over are probably going to be the ones that are recycled and refilled with classical neurotransmitter for later use. Okay, so maybe take a little break from lectures, and there's another one coming.